Our next speaker, Dr. Laura Thornton, graduated from Southwestern in 2009 and is currently a postdoctoral research fellow at the Boys Town National Research Hospital's Center for Neurobehavioral Research. Dr. Thornton earned her doctorate in applied developmental psychology in 2015 from the University of New Orleans. Her dissertation examined the developmental pathway associated with callous-unemotional traits and the utility of such traits and emotional deficits in predicting later antisocial outcomes for youth involved in the juvenile justice system. Dr. Thornton's research interests focus on the developmental neurobiology of callous-unemotional traits, as well as how these traits affect youth involvement in antisocial behavior, interactions with peers, and high-risk behaviors. Please help me welcome back Dr. Laura Thornton.
Hey there, y'all. Can y'all hear me okay? And I'm also getting over a cold, so I don't sound like Kathleen Turner anymore, but I still don't quite sound like myself, so bear with me while I get this going. Thank y'all so much for coming out today. I really appreciate it, and I'm just so happy to be here. And let me see if I can get presenter view going. Is that possible on this one? Yeah. Nope, y'all see that too. Maybe let's not have that. Is that an option? Maybe? Tech person? Can we get presenter view? No. Okay. That's fine. Just asking. You don't know if you don't ask. All right. And I got my thing. So, like Brian said, I'm Dr. Thornton, and today I'm going to be talking about social and emotional neuroscience in youth and focusing on typical and atypical development. And both mine and Dr. Blair's talks are going to be heavier on the atypical side of things. And so my work has really focused on children with severe emotional and behavioral problems, particularly conduct disorder and oppositional defiant disorder. And that's kind of where my experience has been, and I even worked for a year after graduating Southwestern at the Wilco Juvenile Detention Center. So kiddos in the juvenile justice system are kind of my jam, and I've continued to study them and have transitioned from a more behavioral perspective to doing functional MRI work with Dr. Blair. So going from there, kind of what we're going to talk about today is conduct problems and callous-unemotional traits, or CU traits. And then we'll be talking about the role of the amygdala, this tiny little structure in the brain that is super duper important, and kind of how it's playing in a role in the development of CU traits and their maintenance, and really trying to understand how this all comes together. And then I'll talk about the amygdala's role in threat and Anima C processing, and how this may underlie some of the problems that we see in these kiddos. And then we'll kind of bring it all together and talk about kind of the real world consequences for these kind of dysfunctions that we're seeing in youth. So here is a nice picture, pardon me, from Dr. Blair's New England Journal of Medicine paper that shows a variety of issues that children and adolescents experience. And I'll really be talking about adolescents mostly today. So you can see that there's a variety of things there, but my experience is predominantly in the kind of conduct disorder and ODD realm. And this comprises a wide variety of behaviors, a lot of which you can just think of as cruddy behavior from a teenager. So this is rule breaking, this is you know, being truant from school, and kind of reactive forms of aggression, which is I punch you in the face because I thought that you just bumped into me in the hall, and I don't like that. And I may be mistaking the fact that you just bumped into me in the hall for you purposely tried to push me because you don't like me, and so I punched you in the face. Two more proactive forms of aggression, which is punching Dr. Blair in the face and taking his wallet because I want his wallet, and things that go along with that. And CU traits are more associated with these more instrumental forms of aggression. So doing things for gain, particularly. And that's kind of where a lot of my work has focused and what I'll be focusing on today predominantly. So CU traits, I've kind of talked about it briefly at this point, but they have been incorporated into DSM-5 as the with limited prosocial emotion specifier for conduct disorder. So if a kiddo has poor behavior for at least a year and has at least 2 of the following 4 symptoms, they may meet criteria for this disorder. And this is lack of remorse or guilt. And you can kind of think of this as what you see often on the news of people just not feeling bad when they get caught. I don't care that I punched that person in the face. I got their wallet. All of those good things. And a callous lack of empathy. So I don't care that I bruised your face because I punched you in the face, because I don't care. And why would I care? And similarly, being unconcerned about performance. So if I'm doing poorly at school, at work, I don't really have a lot of friendships that are going super well right now, and I'm doing really poorly kind of across the board, and I still don't care. That's that kind of being unconcerned about performance, doing poorly across the board and really not being bothered about it. And then a shallow or deficient affect. So this you may think of as kind of switching emotions on or off, but more of you may not see me being super jovial or happy, but you will see me angry. Things along those lines. So kiddos with CU traits represent an important subgroup of antisocial youth because they tend to be more severe and persistent in their behavioral problems, but they're a pretty small proportion of kids with antisocial behavior. But because of all of these things, they're often more likely to have juvenile justice system involvement, and thus why I've continued to kind of work in this population. And my graduate work focused on the Crossroads study, which was a very comprehensive examination of how the juvenile justice system affects youth over time. And from there, I transitioned to focusing on this kind of neurobiology of CU traits with Dr. Blair and getting to use functional MRI. And this is our MRI scanner from Boys Town. And I'll kind of give a brief overview of kind of what functional MRI does and some fun jargon that Dr. Blair and I will be using for sure. So functional MRI is much like a regular MRI, except it's longer. Instead of just one static picture, we're getting tons of pictures all the time. So as I explained to the kids, I get to take big, beautiful movies of your brain. And this lets me see how your brain is lighting up and using blood, and that shows me what parts of your brain you're using in a different way. And this we can do with functional MRI, and we call it the bold response. So when you hear bold response, this just means we're using more blood in this particular area in the brain. So jargon bit over, hopefully for a little while. The importance of the amygdala. Dr. Blair's theory focuses predominantly on the amygdala for the development of CU traits. And the amygdala is pretty darn important. We use it all the time, just like the rest of our brain. But we often think of it as having to deal with emotions and all of our emotions all of the time, but particularly for processing fear. And it doesn't just do that. It has a lot of other roles as well. So it's not just fear processing or anything like that. It also has a couple of other roles that we'll be talking about today, particularly threat and Anima C. And threat is whether this dog is trying to kill me or not. I may not want to be in front of this dog if it is in front of me in real life. I probably need to move away from that. And your amygdala is involved in signaling that, hey, that thing is coming towards me. I probably don't want to be around it. And then Anima C processing is whether something's alive or not. So is a leaf alive? No. Is this weird Muppet creature alive, or is it my dog Atticus? And I'm fine unless I'm a squirrel, and then I need to worry about it. So you can kind of see how threat processing and Anima C processing go hand in hand, in that I need to be worried about something if it's alive and going to try and kill me. But is it alive and going to try and kill me? So those two things are really going hand in hand, and these represent two different roles of the amygdala in what I'm going to be talking about today. And this kind of going back and forth across from Anima C and threat processing really underlies social cognition. So if you think about what gets us to interact with the world, is it alive? Is it a threat to me? Those seem pretty darn important for ways to go and interact with the world. And I'll be focusing and showing you some data for kind of both of these aspects of the amygdala processing. So we do know that the amygdala in youth with disruptive behavior disorders, so that's conduct problems, so conduct disorder and oppositional defiant disorder, do seem to be disrupted in these youth. And it seems to be a failure to respond to fear and distress expressions. And so this is kind of those, everyone can think of those Ekman faces of the wide, fearful eyes and the kind of very scared faces. Whereas children with disruptive behavior disorders often don't have the same amygdala activation to these fearful facial expressions as the rest of us. And Dr. Blair has shown, and out of his lab, that this is a really negative association when it comes to the influence of CU traits. So not only are kids with conduct problems in general kind of having difficulty with this, but your severity seems to be associated to how much CU traits you have. If you're showing higher levels of these traits, we're getting even less amygdala response to fearful emotional expressions. So we don't really know from here, what does that mean? Does this actually impact anything with the world? Indeed it does. So a former postdoc of Dr. Blair's, Abby Marsh, her lab came out with this and showed that the association between CU traits, so these things that we know are not very great for people, and some real world consequences, that proactive aggression, so that punching you in the face so I can take your wallet and go buy myself a burger, after that is really accounted for by this reduced responsiveness in the amygdala to these fearful facial expressions. So there are some real world consequences that we're seeing for this kind of reduced amygdala activation. So from here, I'll jump into kind of those two different roles that I was talking about, the threat processing and the Anima C processing. So what we were expecting for threat processing and Anima C, so kind of prime you up for both of these, is that we would expect kind of less amygdala response in these DBD youth compared to typically developing youth in terms of threat processing information. Pardon me, and that indeed CU traits would be negatively associated with amygdala responsiveness. So trying to see if these patterns that we've seen in the emotion literature hold here in these other roles of the amygdala. So threat task, you're in an MRI, you're laying down, you're nice and cozy, you have your emergency button ball in one hand that you squeeze if you need to get out, and then you have a remote control in the other hand. And what our participants see is a series of images either coming towards you, so the looming condition, or moving away from you. And this is either threatening or neutral images. So an experience like this, and the participant's job is just to press the button when you see the picture. So this is something kind of like that. So I should have given a content warning, sorry. But I proved my point here, right? Like, thank you. I didn't pay her, I promise. So you have these kind of conditions where things are moving towards you or away from you, and they're either threatening or they're just kind of neutral objects. And we vary the direction, so they're coming towards you or away from you, and we've got those threat and neutral options. So what we can get at here is this amygdala response. And we're expecting that reduced response for our DBD kids. And we get this nice diagnosis by direction interaction in the amygdala. And what you can see here in blue is that whether something's coming towards you or just looming and it's a neutral image, or even a threatening image coming away from you, DBD kids, or pardon me, not DBD kids, because they're not doing it right. The TD kids have all of this activation in blue, whereas our DBD kids are not at all. And you can kind of see the rest of us are having that great amygdala response, and the DBD kids just not so much. I won't pick on you anymore, I'm sorry. And then we really wanted to understand the role of CU traits and how this kind of interacts with this. And what we got here is a CU traits by direction interaction. We didn't find it in the amygdala, but we found it in these other really important attentional regions. So left middle frontal gyrus and left inferior parietal cortex. What we find here is in the red for the looming trials that the higher levels of CU traits that you have, so the farther it goes to the right, the less activation you're seeing in the amygdala, or not the amygdala, these important attentional regions. So it seems like we've got some evidence that things aren't kind of operating the way it should in terms of our hypotheses, that our TD kids are not responding in the same way as DBD kids, and that this does seem to be associated with CU traits. But what does this mean for Anima C processing? Like I was saying, they go really hand in hand and really underlie how we're interacting with our world and social cognition. So Anima C hypotheses, so whether we're telling whether things are alive or not. Expecting less amygdala response in DBD youth compared to TD youth in the amygdala, and again, expecting that it's going to be negatively associated with CU traits. So here we used an Anima C.probe task, and a .probe task is basically where an image is presented, and it's either on the left side or the right side of the screen, and then a dot is presented either on the left or the right side of the screen. Your job as the participant is to tell us what side the dot is on. That's all it is. And what we have here and what we vary is sometimes it's on the same side, so a congruent trial or an incongruent trial. And what our Anima C task does is it has animate and inanimate, negative and neutral, and we just have faces on the animate side. And this really lets us get at this kind of Anima C processing within the amygdala. So nothing jumping out at you, this one. And so what we have here after kind of completing this in functional MRI and thinking about that first hypothesis, so expecting it to be reduced for DBD youth, we get a diagnosis by Anima C interaction. So again, here in the amygdala, what you can see is this bold response. It's a difference score between they're responding to animate to inanimate stimuli. And so what you can see here is that our TD youth are showing a nice big difference between animate and inanimate stimuli. DBD kids, not so much. And this is showing that indeed there does seem to be some processing difficulty. And when we think about the role of CU traits, again, we get this nice CU traits by Anima C interaction, again, within the left amygdala. And what you can see here is just the general bold response, so it's not a difference. And what you can see here is in the red dotted line, that's the average response to animate stimuli for TD kids. But this analysis is primarily done, or only done in our DBD kids, so I'm just giving that for reference. So you can see kind of over at the low level of CU traits that they're pretty much hanging out where the TD kids are. But as CU traits increase, that amygdala activation is decreasing to this kind of Anima C information. And their response and association between CU traits and inanimate stimuli isn't there at all. So what do we kind of take from this? It seems that we do have some more support for atypical Anima C processing in the amygdala for these youth. And this seems to kind of take together that we have not only some emotional atypical responding, but we seem to have atypical responding for threat and Anima C as well for these kids. And this is really similar to our findings with emotion, that the level of CU traits is associated with more severe responses, and that these seem to be particularly important for kind of severity of how your amygdala is responding. And that this amygdala impairment that we think seems to be most strongly associated with CU traits also extends to threat and Anima C, because we really have never looked at this before. So these are kind of some nice kind of new findings showing that not just the emotion processing role of the amygdala does seem to be atypical in these youth. And this has some real world kind of implications. Rachel was talking about the kind of feelings of asociality, and we think this may underlie some of these issues that we're seeing with these kiddos, in that if you don't have the same kind of Anima C processing going on, the signaling of, hey, there's other things in the world that you need to be interacting with, you may not really seek out other people that much. And this is associated with these kind of feelings that kids with CU traits have reported in terms of not being interested in interacting with other people, not feeling super close or warm to other people. And this may kind of underlie that aspect as well. And then threat processing, I think, comes a little bit more naturally to us in this sense, in that if you think that if your threat processing isn't the same as everybody else in the room, you may be a little bit more likely to go take some other risks that other people may not be willing to take. And so you may be more willing to go punch up somebody else to get their wallet because you don't necessarily see that as a risk as much. So these may have some really important kind of real-world consequences for these disruptions and kind of atypical development for these kids. And this really reflects this kind of preliminary step into designing individualized treatment programs. So in general, children with conduct problems and high levels of CU traits are traditionally not as responsive to traditional treatment methods. And this has been kind of widely found, and what we have found to be successful with these kiddos is very individualized to their particular cognitive and emotional style treatment programs that are well-rounded and of course cost a lot of money and all of those things to kind of bring that together and be a bit more holistic in that sense. And so this may represent some of the kind of underlying neurobiology that we may need to be targeting in this sense. So I would like to thank you guys for your attention and from our funders. Thank y'all so much. Questions? President Berger.
The animate and inanimate experiment, I didn't quite understand that. So you have these animate, inanimate, and then you have the threat and the positive. Mm-hmm. Then after the image, you show the asterisk. What is success there? If I get it right, the right answer the right way or the wrong way, how do I measure?
Excellent question. So what's interesting about both of these tasks is that in doing the task, we don't have a behavioral measure of success in that sense. So kiddos aren't necessarily going to be right or wrong in that sense. We're just looking at that kind of amygdala response. So you don't have to necessarily be processing Anima C to get it right, since all you're looking for is that dot. But in able to show all of those images, we're able to generate that amygdala response, and that's what we're going for in that aspect. Does that answer your question?
So the aspect really is kind of a red herring.
Yeah, a little bit. It shows us, it gives us a particular measure and thing that we need have kind of so that we can tie the bold signal to something. So excellent question. Mm-hmm. That's an excellent question. So I forgot to rephrase questions. Is that, so let me just pull up the slide. The question is that in this one? All right, so here what you're looking at, and the question is, why is it positive for the TD kids and negative for my DBD kids? And the reason that is that this is a different score, so their animate response is greater than their inanimate response. And then what that means for the DBD kids is that their response to inanimate was a little bit greater than their animate response, but what you can see there is that the error bars cross 0. So it's not going to be very different from each other. But the main point is that the DBD kiddos are indeed not responding the way that the TD kiddos are. Excellent question. Dr. Gracci.
Are they're collapsed across neutral and negative?
Mm-hmm.
So there was no separation or difference between their amygdala response when it was a negative versus a
We didn't get any emotion findings in this one, and that is a kind of a odd thing about this one is that we didn't get any emotion response at all, even in our TD kids. So we think that's likely a paradigm problem. So indeed, excellent question. Any other questions? Yes, sorry.
We had a statement related to impairment in amygdala responding, how that might be linked to CU traits. Right? And one of my questions I have is when I think about amygdala responses is, and you also mentioned that biology, the neurobiology is something we should also take into consideration. Has mirror neurons ever come into the conversation related to the unemotionality of responding, and thus that you're not receiving some activation from your neurons, or you're not picking up on and feeling some empathy? Has that been a discussion at all within?
That is an excellent question that I will totally punt to Dr. Blair. Because indeed, I've only made this transition in the last 18 months, so I don't know the literature quite as well as Dr. Blair yet, and that my understanding, not particularly so.
For a while, the mirror neuron story was, people were pushing it as a potential route into problems here. But the mirror neuron story in some respects has fallen to pieces. We do have, so these things are mirror neurons. So, you know, if I do this movement, there will be neurons that will fire in my brain when I'm doing that movement. If I see you do this movement, those neurons will fire as well. But and people sort of made a case for emotional responses being similar, but it really has not stood the test of time. I mean, those neurons that you're seeing in the amygdala are not mirror neurons. They're neurons that fire when emotional stimuli come in, and they're neurons that fire when you've learned about cues that are associated with those emotional stimuli. They're not, they're not mirror neurons as such. So, mirror neurons through is a very different brain area as well, which really don't play into here as well.
Thank you for saying that. Yeah.
So I'm curious about your measurement of the CU traits in particular, and how confident you are about the validity of it.
That is an excellent question, and that we predominantly use the inventory of callous-unemotional traits, and this was developed by Paul Frick, who I did my graduate work with. So there's a strong body of evidence showing that the measurement of CU traits does seem to be particularly useful, and in that it shows you kind of more information when you're looking within antisocial youth. If you're looking in the general population, it's not going to tell you much in terms of antisocial behavior or predicting lots of stuff. Impulsivity is going to tell you who's going to be the worst at the end of the day. However, if you're looking at within antisocial youth, CU traits seem to be particularly important for distinguishing an important subgroup of youth who have this more persistent, severe, and violent forms of behavioral problems. So this was all done at NIMH initially, and we're continuing to use the ICU at Boys Town, and it's always administered either self-report or parent report, and we predominantly use parent report.
So in determining that last part, are you identifying kids that have the disorder but virtually no CU traits, and then kids who have the disorder and high CU traits? Are you able to find groups that are...
We are able to do these kind of high-low group analyses and look at it more continuously. All of these analyses are ANOVAs and ANCOVAs, so we're looking at it more continuously. But we have been able to do high and low groups successfully as well. But yeah, when they're coming in for participation, they do get diagnosed by a psychiatrist as well. But we tend to also get more behavioral measures, so things along the lines of the child behavior checklist and kind of these more broad assessments of different levels of behavioral problems as well. Excellent. Yes?
So, looking at the amygdala difference in some of these other ones, it's like heightened, right? We're looking at typically developing, and then lower for these in terms of the behavioral disorders. Is there any sense that this is like normally distributed? That there might be like kids on the other end, for instance, who have a even more, heightened response, and the baby maybe more anxiety or something like that?
That is an excellent question, and is kind of a hot topic right now in the field, is trying to determine whether the root of CU traits is this amygdala responsiveness in particular, and where trauma plays in. And Dr. Blair is totally going to touch on that a bit more, so I don't want to steal his whole talk. But indeed, we do find that most kiddos with conduct problems do tend to be more bothered by their behavior, the effects that it's having on other people, often have high rates of anxiety and other comorbid issues. And we do have some initial evidence that, yeah, they may be having this kind of increased hypersensitive amygdala response. So I think that's all I have time for, though. Come talk to me.